Ileana obtained dual degrees in physics, one from physics from École Polytechnique in France and in medical physics from McGill. She then went on to earn her PhD in physics from Université Paris-Saclay, with a doctoral work focused on functional and diffusion MRI microscopy at the single neuron level. She then did a postdoc fellowship at NYU and the Center for Biomedical Imaging at EPFL, working on combining diffusion MRI with biophysical modeling for both clinical and preclinical applications. Ileana is currently an SNSF Excellenza Assistant Professor and is the PI of the ERC Starting Grant in the Radiology Department at Lausanne, the Lausanne University Hospital. Research spans theoretical biophysical modeling, experimental design and validation, and clinical translation to neurodegenerative and psychiatric diseases. She's the recipient of the Jean Timmons, Costello, and Irtelis fellowships, and her research work as well as her educational courses have received multiple excellence awards from ISMRM. So today she's going to talk to us about some interesting work looking at dynamic fluctuations in the apparent diffusion coefficient, ADC, due to microstructural changes underlying neuronal activity, which she calls ADC-fMRI. Please join me in welcoming Dr. Ileana Jelescu.
Hi, everyone. So thank you very, very much for this invitation. It's really a pleasure to be speaking to you today and with you today. So, well, I'm sorry I cannot be in person. We're already juggling some time zone difference here, but it's really a pleasure to be introducing to you this topic that is dear to my heart of ADC-fMRI. And well, I guess I'm quite flexible with time, so maybe if there are questions even during the talk, you can feel free to ask. You don't have to wait until the end, but just maybe speak up because I may not see if you write something in the chat. So just unmute your microphone and feel free to shout out. Okay, so um, okay. So basically, the premise of the talk is that there are some intrinsic limitations of bold functional MRI contrast and of other vascular contrasts, which that they necessarily rely on this indirect neurovascular coupling. So the way the sequence goes, I think most of you are probably familiar with it. If you have an increase in neuronal activity, you also this is followed by an increase in blood flow and volume. And so you have a change in the ratio between oxy and deoxyhemoglobin in the venous blood. That basically means your venous blood becomes more oxygenated due to this neuronal activity. And so this will make a more homogeneous field strength around your blood vessels, more homogeneous magnetic field. And so the T2 or the T2 star will become longer. You will increase your signal. And so this is called the so-called bold signal or blood oxygenation level dependent that gives rise to a positive signal increase upon brain activity. And so you can see here that this is a bit of an indirect effect because if we have a stimulus here at a certain time, the bold response is slow and also a bit sluggish and spatially not so specific. So you start with this initial dip and then you have this peak where basically it maximizes after 5 or 6 seconds after the stimulus. Takes a while to come down, and then sometimes you also have this post-stimulus undershoot. So this response basically is shifted in time and can also be shifted in space because the veins that generate most of the bold signal may not be in closest proximity to the area of the actual site of neuronal firing. So this is basically an indirect detection of neural activity that gives rise to limited spatial and temporal specificity, and particularly for our interest today, also reduced sensitivity in the white matter. So why is that actually? White matter activity is actually a blind spot for almost all of the current neuroimaging techniques. So it's very hard to see information flowing through the axon fibers. With EEG or MEG, for example, we are limited in depth by the sensitivity, so it's hard to go anywhere beyond the cortex. And for functional MRI that relies on this neurovascular coupling that I introduced on the previous slide, the sensitivity is rather limited by vasculature. And so indeed, white matter is much less vascularized than gray matter. So we don't have this very strong hemodynamic response like we do in gray matter. So there is altered vasculature, but also the energy requirements are different. And so this very different hemodynamic response in the white matter basically obscures a bit bold imaging. And if you try to look at white matter bold studies across the literature, they are not as simple as just doing your usual GLM, right? Typically, you have to tailor the analysis methods specifically to the white matter activation. So here you can see the response timing of bold in the white matter is very, very different between superficial white matter, which is still has a lot of partial volume from nearby cortex, etc., as compared to middle and very deep white matter. You see the timings here are extremely different. And so basically you need a different response function for different brain areas, and it makes the analysis complicated. But the motivation behind it, I mean, why would we need to even access what happens in terms of white matter traffic? I would argue this would be very useful for things like neurosurgical planning. So instead of just relying on tractography reconstructions, you could also see, where the information is flowing to try to spare the really relevant fibers, or for things like stroke rehabilitation, where sometimes you also have a significant portion of the white matter that is affected. And then it kind of begs the question, is that white matter still functional and able to transmit information, or is it just there as a prop, but it's actually not, there's no traffic flowing through it anymore? So I think it would be actually very, very relevant to try to see what kind of information is flowing through the white matter. And so definitely we want to use a different contrast than a vascular contrast for this purpose. And so this is where we turn to diffusion. And so this is just one slide on diffusion MRI in general. I hope most of you are familiar with it, but basically diffusion MRI gives access to what we call the mesoscale. And so in an unrestricted homogeneous medium, such as this glass of water, we know that the probability of displacement follows a Gaussian or normal distribution. But in biological tissue, the random walk and this diffusion of water molecules is actually heavily affected by the cellular layout and the membranes that act as restriction or hindrance to these water molecules. And so this displacement probability function is no longer a Gaussian, typically tends to be more peaked because basically you have a higher probability to encounter molecules closer to where they started than further away, because if anything, the membranes just again act as a restriction or hindrance. And so very much like this drunk person trying to make their way home and like bouncing on the lampposts and walls, the water molecules, basically they encounter these, the cellular layout and the membranes, and their path somehow also encodes information about what kind of cellular layout they encounter. And so this gives rise to this kind of super resolution effect of diffusion MRI. So we know that the image spatial resolution is typically very coarse. So we're looking at 1 to 2 millimeters at best in the human brain, while the cellular features, the cellular layout and the typical sizes and distance between them is rather on the order of a few microns. And so this is where we are lucky that basically in a diffusion MRI experiment, the path that these water molecules travel is also on the order of a few tens of microns. And so the path of the water molecules can actually encode information about what the cellular layout was like at the micron level, and basically at a spatial scale that is much, much higher than our image spatial resolution. So diffusion MRI is really an excellent probe of tissue microstructure. And so what does this have to do with looking at white matter activity? So the idea was basically to try to use this diffusion MRI contrast as a functional contrast in a very specific way. And so today I will first go through this brief history of diffusion functional MRI, because it's not something I've invented. It's been around for a while. But then I'll also present to you our more recent efforts to try to minimize bold contributions to the diffusion fMRI signal and really have something that is heavily weighted towards diffusion microstructure and no longer towards the vascular response. And then we're trying to see how we can achieve uniform sensitivity to this microstructure fluctuations that are functional using isotropic diffusion encoding, and this is a very strong value for white matter, of course. And then the last part is something more in development, whether we can even go beyond fluctuations in the apparent diffusion coefficient and actually look at functional changes in what we call kurtosis that I will also describe at that point, and membrane permeability. But let's start with this brief history of diffusion functional MRI. So as I've described, the neurovascular coupling relies here on what happens basically to blood oxygenation in the veins during neural activity. Whereas for diffusion functional MRI, we want to rely on something that we call neuromorphological coupling. And what this means is that basically it's been shown with high resolution optical techniques that when action potentials go through, there is some kind of transient membrane displacement and mild swelling and coming back of the neuron during these action potentials. And so these displacements, they occur at the level of the neuron cell body here, but also in the dendrites that lead off from this neuron. And actually, this has also been shown to occur in myelinated axons. So here you have this transient displacement of the myelin sheath as action potentials are firing through the axon. That was shown with spectral interferometry. And what was interesting to us is that they actually showed that it's really the myelinated segments that swell transiently and then come back to baseline rather than the unmyelinated parts at the node of Ranvia. But so this transient deformation or swelling of the cells during neural activity is something that's kind of not controversial in the sense that it's really been shown with optical techniques. And so because diffusion MRI is so sensitive to this cellular layout and what the cells and their environment looks like, there came the question, can we use diffusion MRI as a functional contrast? Would the diffusion weighted signal, for example, change to pick up these mild displacements of cell membrane and then coming back to baseline? And so this, as you can see, was proposed, 25 years ago, actually, by the group of Denis Le Bihan. And so here I just want historically to go back to this first ADC-fMRI experiment. I think this was really a very good and solid paper in the sense that they really took care already of a lot of the confounds that can be reproached. And so this was called transient decrease in water diffusion observed in human occipital cortex during visual stimulation. And they really calculated, so they were acquiring interleaved B values as a time course. So volumes with a B value of 200 and 1400 interleaved as a time series. And then from each pair of B values at a given time point, you calculate the apparent diffusion coefficient. And so you get a time course of apparent diffusion coefficient. And this was the cluster that showed a decrease of apparent diffusion coefficient during visual stimulation with the matching bold response underneath. That is, of course, much more widespread. But the goal with this technique initially was to look still in the gray matter. So at that time, ADC-fMRI was not about white matter, it was about gray matter, but trying to get a cluster that is spatially and temporally more specific to the activity than bold itself. And so this is what they were trying to do, that basically the ADC drops immediately, whereas the bold response actually takes a longer time to shoot up. And so, unfortunately for this method, the following papers just started to raise some controversy. And the reason for this is that in the follow-up paper a few years later, basically they gave up on this idea to calculate an apparent diffusion coefficient at each time point from pairs of diffusion-weighted volumes, but rather they just decided to look at the diffusion-weighted signal, okay? And just focus on the fact that basically you see that the response here with B1800 is again faster than it is with the bold that serves here as a reference. And so basically they were saying the fact that we introduced diffusion weighting already shows that we have a different kind of response here early on that kicks in much sooner. But what they omitted to maybe emphasize is that if you look at diffusion-weighted signals, basically, because it's a spin echo design, you also have T2 weighting. And so this T2 weighting of the diffusion weighted signal by design makes them also intrinsically bold weighted, right? There is such a thing as T2 bold. So if you have T2 weighted time courses, they will also contain some bold contrast. And so this is where this was basically immediately proven to basically also be heavily affected by hemodynamic effects by a paper by the group of Carla Miller at the time. So these were like back-to-back publications in PNAS where they really did. So they repeated the visual experiment of the group of Le Bihan, showing the same things, like there is a response, of course, in these diffusion-weighted time courses, but then also just during hypercapnia. So excess CO2, it really induces vasodilation without any sort of without any sort of neural activity underlying it. Basically, they showed that the diffusion-weighted time courses also showed a response in the case of hypercapnia because you have this T2 weighting of the diffusion-weighted time courses, and therefore you have bold really interweaved with this diffusion weighting necessarily. And so this became problematic, and people started also to understand that we really have to work harder if we want to do ADC functional MRI to have time courses where we really try to keep bold contrast out of them. So if we want bold, we're just going to do T2 star or T2 weighted. But if we want diffusion, contrast, and microstructure sensitivity to neural activity, then we want time courses that do not contain any sort of vascular contribution to them, because otherwise they are just interwoven with bold contrast. And so, nonetheless, when you work very hard to remove any sort of vascular contamination, you can find this effect, which is measurable and exists. So people have shown, for example, that the drop in ADC can be detected in the absence of any vasculature. So this was in the mouse optic nerve that was very easy to isolate and was not contaminated by any blood vessels. And so parallel to this optic nerve, there was no change in ADC between baseline stimulus on and stimulus off. Whereas in the perpendicularly to the optic nerve, so to the fibers, there was this drop in ADC during stimulation and then back to baseline when you turn the stimulus off. So this was a very nice experiment. Then again, the group of Denis Le Bihan also showed a few years later that with chemical disconnection of the neurovascular coupling, so using this nitroprusside, the diffusion fMRI response was unaltered, whereas you heavily suppressed, of course, the bold response, because now there was no vascular response to the neural activity going on. And then from the group of Noam Shemesh in Lisbon at the time, using ultra-high temporal resolution line scanning, they also showed that this drop in ADC basically occurs way before the bold response kicks in. So we really have this temporal dissociation of the two and much sharper response of the ADC drop compared to bold that is always heavily delayed. So these experiments were really very encouraging. It just means that we cannot work with diffusion weighted signals because they're intrinsically T2 weighted. We have to work with time courses where we try to eliminate as much of this vascular contribution and bold contribution as possible. And so we've spent quite some time trying to understand how we best minimize these bold contributions to diffusion fMRI. And so in order to explain those, what are first the possible sources of bold contamination to this method? So if you take a voxel here between rest and active states, you see that there are blood vessels everywhere, right? So you go from rest in an inhomogeneous field to active where the field becomes more homogeneous because the veins become more oxygenated. And so the signal that you measure is, well, T2 weighted because of the spin echo design, and it's also weighted by your diffusion weighting here, the B value, and the ADC of the tissue. And so in order to eliminate most of this T2 weighting, what we do is that we move from the signal to the ADC by taking a ratio of two signals with two different B values acquired in interleaved fashion. But then another problem is that even this diffusion weighting here, the B value, I mean, it's largely driven by your diffusion gradients that you impose in your sequence, but you also have this cross term with background susceptibility gradients. And you see that these susceptibility gradients, they change a lot with bold-like effects, so with this venous blood oxygenation. And so basically your B value, your effective B value changes between oxygenated or deoxygenated blood. And this is also a problem. So in order to reduce this cross-term contribution here to the diffusion weighting, it's better to work with things like twice refocused spin echo sequence designs. And finally, here, if we break down our voxel into basically tissue, blood, and cerebrospinal fluid, we see that in the active state, so you have an increase of blood volume, but it is usually absorbed by a decrease in cerebrospinal fluid. And so the ADC that we measure is nonetheless some kind of weighted average between these three compartments. But what we care about is really this tissue ADC. So we also have to suppress as much as possible this direct contribution of the blood pool and the CSF pools to our measurement. So the blood pool is a bit easy to get away with. So here we need to acquire pairs of B values to calculate our ADC that are higher than, for example, 200 seconds per millimeter squared to stay away from any fast flowing spins that our actual perfusion will be already heavily suppressed with this B value. The CSF is harder to suppress. We actually, for now, just decided to live with it because the best way to do it would be to do some kind of inversion recovery preparation to null the CSF signal, but then you're really also losing some SNR there. So this is a bit of a problem. So basically, this is how an ADC-fMRI experiment looks like. So we have to go with twice refocused spin echo sequence or something that will kill as much as possible cross terms with background gradients. And we acquire a series of interleaved B values. So for example, 200 and 1000 here. We use pairs of such images to calculate an ADC at each time point, and then this becomes our ADC time course. And of course, for bold, I mean, it's a traditional bold design. You just acquire either spin echo or gradient echo API, whether you want to do T2 bold or T2 star bold. But you really need to indeed look at ADC time course to minimize the T2 weighting, choose the B value pair carefully to minimize direct blood contributions, and work with sequence design that minimize cross terms between our diffusion gradient waveforms and the background gradients from the magnetic susceptibility sources. But if you do this, basically you can detect very clean ADC activation clusters. So here this was a visual and motor task that we did in the human brain at 3T. And so you see in the visual cortex, you have very largely a negative ADC response here to the task, whereas the bold is, of course, a predominantly positive bold response, but you can see that the ADC response is very much synchronized with the task, and the bold is really quite delayed. And of course, we had similar findings in the motor cortex. And what was interesting to us looking at these clusters of activation, so the orange here is the bold and the blue is the ADC. And the ADC clusters also seem to follow some kind of path in the white matter. And so this really triggered or piqued our interest in terms of saying, okay, a lot of people have been focusing on alternative functional MRI contrast for gray matter, but what actually, if actually we could use ADC-fMRI contrast as a way to look at white matter activity. And so we started looking more closely into these ADC time courses a bit everywhere in the brain. And basically, you can also derive this ADC connectivity between different brain regions. So this was during a visual task. So of course, the connectivity of the visual system is very strong here. And you could see that hierarchically it was clustered separately from the rest of the brain. But so then if we zoom in on this visual cortex connectivity here, we found actually really very clean hierarchical organization of this connectivity between the different visual areas and the white matter that connects to this visual cortex. So we know that for the visual system, the area split between a dorsal stream and a ventral stream. And the connectivity that we found through these ADC time courses exactly reflected that. So we had here a separate branch for the primary and secondary visual areas here, V1, V2, that were clustered together. But then we had moving on from this, we had a very nice separation between this dorsal stream here, the ventral stream with V3, V4, and then the ventral stream, the dorsal stream here with V5 that were separate, that were really as expected based on this organization. And also the ADC connectivity also brought in together these white matter areas, so optic radiation and the IFO and ILF that are really part of the visual system and that also were also clustered with the primary and secondary visual cortex. So this really showed that there was some logic to the way these ADC time courses were synchronized together, and that we could really retrieve this organization of the visual system very nicely through these time courses. And so then, because here we were looking at connectivity, although this was essentially a task fMRI design, essentially, we also started to wonder, but can we actually even just look at resting state using ADC-fMRI time courses? And so we tried this with our optimized protocol to minimize bold contributions to ADC-fMRI. And so for resting state functional connectivity, basically, we did this RY to RY connectivity where you parcellate the cortex into different regions and then you look at their Pearson correlation. But this time we're also able to augment basically this matrix, not just with gray matter nodes, but also with white matter nodes. So taking the white matter not just as an edge anymore, but really a node of its own from the JHU atlas. And so basically we have an extended functional connectivity where we have some edges that are gray matter to gray matter connections, other that are gray to white, and other that are white to white connections. And so you can see that for the gray to gray matter connections that are here in green, basically between bold fMRI and ADC fMRI, the connectivity is very well preserved. But what we saw with ADC fMRI in addition was actually much stronger or cleaner connectivity of gray to white matter and white to white matter areas. And this is because we think we have better sensitivity to what happens in the white matter with ADC fMRI than we do with bold, because again, bold relies on vasculature and vasculature is very scarce in the white matter. And so we also derived some of these graph metrics based on this connectivity. So here looking at average node strength, clustering, and global efficiency. And so for gray to gray matter connectivity, this was quite similar between bold and ADC. But again, for white matter to white matter connectivity, the ADC fMRI graph metrics were higher than for bold. So higher node strength, clustering, and global efficiency. Now, if something is higher, it doesn't mean it's better. So this is just to show basically that the graph structure was also different with ADC fMRI than it was with a bold fMRI. So including these white matter nodes yields very robust information that is actually not matched by bold. But to really understand what this means, this is still something that we're investigating at the moment. Okay, so this was our early work on ADC fMRI, really just trying to mitigate bold contrast and starting to look a bit at what happens in white matter. But then we really spent more time to really optimize this protocol also for white matter. And there, what do you need to do? So basically, I'm showing this experiment of Space and colleagues again in the mouse optic nerve, where I was trying to show that basically perpendicular to the optic nerve, you had this drop in ADC. So if you apply diffusion weighting perpendicular to the nerve, you have this drop in ADC and then coming back to baseline. Whereas if you apply diffusion weighting parallel to the optic nerve, you don't have any effect. And so basically you see that you also have this anisotropy of response in the white matter because of these microscopic fluctuations, you have better sensitivity and you pick them up if you encode for diffusion perpendicular to the bundle than if you are parallel to the bundle. And so then it means in the human brain, I mean, white matter fibers, they're oriented in all sorts of ways. So if you want to look at global white matter activity in the brain at once, you cannot choose a single direction. I mean, you can make it optimal for corpus callosum and then be perpendicular to corpus callosum, but you're necessarily going to be parallel to other areas where there you will really have reduced sensitivity to this effect. And so the questions for us was, how can we achieve isotropic sensitivity? And so people in the literature, people have tried this before by taking like 3 orthogonal directions and averaging, etc. But then here you really, really lose on temporal resolution, essentially. And so instead, we moved away from this and we exploited this new way of encoding for diffusion, which is no longer so-called linear. So having these two diffusion gradients here of opposite polarity that basically achieve diffusion encoding in one direction at a time, that is the vector resultant of these gradients, but rather turning to these advanced gradient waveforms that basically achieve isotropic diffusion encoding in one shot. So in a single diffusion weighting, you basically weigh two displacements in all spatial directions at once. And this was largely proposed, not for fMRI purposes at all, but it was proposed by the group in Lund originally. So this was a lot of work from Daniel Topgaard, Marcus Nilsson, and Philip Stepankiewicz. And so we now implemented this isotropic ADC-fMRI to try to see if we are better at capturing white matter activity in this way. And so we tried to compare the responses between linear encoding of ADC-fMRI and spherical encoding. And so you can see here that relative to some white matter fiber, depending how you apply your diffusion gradient with the linear encoding, you expect more or less response, or stronger or weaker ADC response, whereas with spherical encoding, you should be immune to this. And so we compared in a visual task between linear ADC-fMRI, which is here in orange, and isotropic ADC-fMRI, which is blue. What was the density of significant voxel or responding voxels as a function of the angle between the fiber and the diffusion encoding? And so you can see with linear encoding, you have higher voxels that were significant, where basically the encoding was perpendicular to that white matter orientation locally. Whereas with isotropic, I mean, you do have this slight dependency here, but essentially it's much more flat. And then in terms of the ADC percent change, with linear encoding, of course, the ADC response was more dramatic when you were perpendicular to the fiber than parallel to it. Whereas with isotropic encoding, it was weaker, but at least it was stable or immune to the orientation throughout. And this kind of dependence we also reproduced very, very nicely in our synthetic substrates that we grow for white matter, where we made them also swell mildly and then replicated both this dependency of the ADC change with the orientation of the linear encoding with respect to the axons in the linear case, and the absence of a dependency with isotropic encoding. So this was then validated with simulations, basically. So now, keeping this isotropic encoding for doing ADC-fMRI in the white matter, here you can see, well, sorry, we can see still for a visual task the responses to with linear encoding and isotropic encoding, and then the comparison with the bold response, which is much more sluggish. So here in blue, you have the ADC response, and the colored ones are the diffusion-weighted signals, the B200 and the B1000 respectively, and then orange here is the bold. And if we look at spatial maps of active clusters with this visual task, we see that, of course, with bold, we get this typical visual cortex that we're very much used to seeing, and the thalamus here that acts as a relay. Whereas with ADC-fMRI, basically the clusters of significant areas that survived the group level comparison were really actually along the optic radiation here that is depicted in pink. So basically, isotropic ADC-fMRI really yielded white matter activation clusters that were not matched by bold clusters here. You see the bold tends to avoid this optic radiation, whereas the ADC is really kind of aligned along this track. So this is why we say that they are really complementary. The goal is not for ADC-fMRI to replace bold, but rather to use it in white matter, where it seems to have more of an advantage. And so we also did some of these experiments in the rat brain on our 9.4 Tesla system at EPFL. And so here in the rat brain with visual stimulation, basically there is a difference in bold response between 1 hertz flickering, which is very slow, and so it's really perceived as different flashes, versus 25 hertz, which is above this flicker fusion frequency and then is perceived as a continuous stimulation. And so you see with the 25 hertz, you have a lot more negative bold in the lateral superior colliculus and the visual cortex, whereas with the 1 hertz stimulation, you basically have this massive positive bold response in the superior colliculus. And so looking at ADC-fMRI responses with a similar kind of paradigm, first of all, the maps were not so different between the two frequencies. We're trying to understand what is kind of the negative bold equivalent of ADC, but there seems to be almost none. So here you can see you have your positive response in the medial superior colliculus with a mild, this is bold, here with a mild ADC decrease. But what we found interesting was that in the corpus callosum here, again, in the white matter, there was no bold response whatsoever, whereas we indeed also detected a drop of ADC in the white matter upon this stimulation. And so here the take-homes were that the isotropic ADC-fMRI detected negative ADC response, mainly here in the medial superior colliculus. The ADC response was a bit less robust to vascular confounds in the lateral superior colliculus. So here where you really have a lot of vasculature at high field, we still found this positive ADC, which we believe is actually really just a bold contamination. But nonetheless, in areas where there was no bold response, namely in the white matter, ADC-fMRI did detect activity there, which means there is also some complementary and genuine contrast to it. But it's certainly harder to do ADC-fMRI at a higher field because of necessarily more sources of susceptibility and bold contamination. And so then coming back to these experiments with hypercapnia that basically killed diffusion fMRI a few years ago, we also wanted to reproduce this to see if with our protocol that is supposed to be as immune as possible to vascular contributions, we're also immune to hypercapnia-driven vasodilation. And so basically, if there is still a vascular contribution to ADC-fMRI, we should see an association with respiration in the absence of any underlying neural activity. And so we did this breath hold experiment, acquiring also time courses for multi-echo grade and echo bold, but also our ADC-fMRI with isotropic encoding. And then we did these cross correlations between the end tidal CO2 signals and each of the time courses. And so what we found is, of course, very, very strong correlation of bold fMRI with end tidal CO2. There is still strong correlation in the diffusion weighted time courses because they are T2 weighted. But we found basically near absent significant correlations of end tidal CO2 with ADC-fMRI time courses. If you find anything, it's really in the CSF here, where we know there is some absorption of the changes in blood volume are taken up by CSF volume. And so we believe this is why we see this in heavily ADC contaminated voxels, sorry, CSF contaminated voxels. And so just to make sure that the absence of correlation here didn't come from the fact that the CNR or SNR of the ADC, the time courses is lower than it is with bold, we also amplified the noise of the bold time courses to match the sensitivity or the SNR levels of ADC. But even for noise levels that even exceeded the noise levels we had in ADC-fMRI, we could still find this very strong correlation of bold time courses with end tidal CO2 that were not matched by ADC. So this really means it's not a sensitivity effect. It's really that the source of the contrast that is vascular is no longer there in the ADC time course. And then very recently, so this is not really published work, but it's going to be presented at OHBM later this year. We wanted to come back to this question, is there an ADC equivalent of negative bold? And so for this, we started doing interleave TMS fMRI, so transcranial magnetic stimulation. And so if you apply TMS pulses at a certain frequency and only 90% of the person's rest motor threshold, basically you can induce very controllable negative bold in the contralateral motor cortex with no positive bold response in the ipsilateral motor cortex because you're subthreshold. So you're not really triggering an involuntary hand motion. You're under that threshold for firing all the way up to the limbs, but you're perturbing it enough that it sends a suppression and a negative bold in the contralateral M1. So this was shown by Shimomura and others a few years ago. So this is a fairly established negative bold paradigm that we then also replicated. So we had this TMS coil that could go inside the MRI scanner with the dedicated MRI coils that were compatible with this. And so we had this paradigm of interleaved TMS and MRI acquisition, where we did both bold fMRI, but also our ADC-fMRI time courses with the usual design of two interleaved B values and then calculating ADC at each time course. And so basically, these are the group levels analysis results of this TMS experiment, where we find, as expected, contralateral negative bold in M1, and also in these diffusion weighted time courses that are T2 weighted, we also find this negative response in the contralateral M1. So this is really according to previous literature, nothing surprising there. We basically found no ADC response in contralateral M1 whatsoever, neither positive or negative. So would this mean that there is no ADC counterpart to negative bold? This could be. Or again, here the question is still, do we just not have enough CNR? And so here we don't really have an answer. I do think there is a lot less source of contrast for negative bold equivalent in terms of neural fluctuations or reconfiguration, but we don't have for now direct proof of this. What was interesting to us is that on the ipsilateral side, so there was no bold because we were subthreshold, sending subthreshold TMS pulses, but we picked up this positive response in the B200 time course. And so we're wondering if combining this diffusion contrast with T2 contrast, we're not getting enhanced sensitivity to something like subtle neural activity or activation. So it's something that is low enough not to trigger the full-fledged response and involuntary hand motion, but still something going on that could be picked up with our diffusion contrast. And we also have found this tiny, this tiny positive ADC cluster in the ipsilateral M1, but closer to the interhemispheric area that we're still working on interpreting, basically. And so for the few minutes I have left, I just wanted to present this other experiment that we did, trying to this time go beyond fluctuations of apparent diffusion coefficient with neural activity, and trying to see if we can also detect functional changes in kurtosis and so-called membrane permeability. So just a brief recap, what is kurtosis? So I explained basically that the displacement probability function is no longer Gaussian, it's more peaked. So this means that beyond a certain B value range, so here above B1000, basically we leave this Gaussian approximation that would give us a linear relationship between the log of the signal and the B value here. So you see there is a departure from this Gaussian behavior. And the fact that this is more peaked means there is positive kurtosis. So you start to have this curvature. And so this curvature can be captured by a second order term in B squared. So this basically now instead of fitting a linear function, you're fitting a parabola to your second order polynomial to the log of your signal. And so you can estimate kurtosis from this. And so kurtosis can be used, diffusion kurtosis imaging is an extension of diffusion tensor imaging in the microstructure world, where you can also look at things like, so here you have mean diffusivity from DTI and mean kurtosis from kurtosis imaging. And so this kurtosis is basically a measure of the variance of diffusion coefficients in your voxel. So it's a measure of the heterogeneity of the voxel in terms of different diffusion domains. And it's also a measure a bit of complexity. And we see that if we have permeability here, so water fluxes across the cell membrane, basically at very long times, everything will be mixed and homogeneous again. And so the kurtosis will go to zero. So you also have this time-dependent kurtosis here that gives you a good measure of the exchange time between the inside of your cells and the extracellular space. And so in the same way that people have shown basically that we have this drop in mean diffusivity or apparent diffusion coefficient with activation due to these transient enlargements or fluctuations of the cell membrane, Ruliang Bai and colleagues also proposed that because you have this active water cycling through the pumps that decreases when the neural activity is altered, we're wondering if this change of permeability or of water fluxes with neural activity could be picked up, by something like mean kurtosis functional MRI. So looking at how cell membrane permeability is affected by this neural activity. And so we tried to do this in a task fMRI block design in the rat brain. So again, these blocks of rest and stimulus using forepaw electrical stimulation in the rat. And so we had to measure fast kurtosis with a subset of measurements. And so basically we couldn't get a full kurtosis estimation in just one block. So what we did is that we distributed all the measurements we needed in the rest condition across all the different rest blocks and all the measurements we needed for the stimulus or active condition across all the different stimulus blocks. So we don't have a time course anymore. We just have a measurement of kurtosis at rest and during stimulus as a result of this 10-minute kind of paradigm. And so we have a measurement of mean diffusion at rest, mean kurtosis at rest, mean diffusion during the task, and mean kurtosis during the task. But then we do this at different diffusion times to also be able to estimate this time-dependent kurtosis and the exchange time. And so these are the results. So as mentioned, we were doing electrical stimulation of the forepaw in the rat. So we expected response in the contralateral somatosensory cortex. And so here, of course, we found this very characteristic positive and strong bold response here in this somatosensory cortex. And interestingly here, the diffusion counterpart of that was that we measured a dramatic decrease or significant decrease of mean diffusivity in the active area in the somatosensory cortex. This was expected because we know there is a drop of ADC during neural activity. And we also measured an even more pronounced decrease of mean kurtosis in the somatosensory cortex. Now in the control region here, anterior cingulate cortex, there was no bold response. This was also expected, and we also measured no change in mean diffusion or mean kurtosis. So this is kind of a sanity check. But then what was interesting is that in some subcortical areas here, like the thalamus, so with our protocol design, we didn't have enough sensitivity to pick up any bold response in the thalamus. It doesn't mean it doesn't exist. It just means with our setup, we were not sensitive to that. But remarkably, although we couldn't measure a bold response, we were still able to measure changes in diffusion and kurtosis. But this time we measured an increase of diffusivity during activity and an increase of kurtosis. So the sign of the change was opposite to what was happening in the primary somatosensory cortex in this thalamus relay area. And so if we look then at different brain regions, so we have primary somatosensory and motor cortex, secondary motor cortex, but then you also have these subcortical areas, thalamus, hippocampus, and striatum. Basically, we found a bold response here in the cortical areas. But the diffusion response in kurtosis, so we have a negative change in these primary cortical areas here in mean diffusion and kurtosis, but we really found overwhelming increases in the subcortical area. So again, a change in sign of the response that was very interesting and is worth investigating further. And then this time-dependent kurtosis, so looking at the kurtosis as a function of diffusion time and fitting this cell membrane permeability or exchange time from it. So this was not significant, so please take it with a grain of salt. It's just that qualitatively, this exchange time basically decreased between rest and stimulus in both the contralateral and the ipsilateral area. So this means that basically during stimulation, it was more permeable than at rest. And this is also what we would have expected. So it kind of goes the way we were anticipating, but since it was not significant because the error bars here are very large, unfortunately, we cannot overinterpret this finding either. But it seems we have complementary underlying mechanisms for bold fMRI versus mean diffusion and mean kurtosis, because in these subcortical areas, it really behaves very differently. And we have an increase of diffusion and kurtosis in secondary somatosensory cortex and subcortical areas, which do require further investigation, whether this is coming from an different inhibitory excitatory balance or glial versus neuronal contributions. We're not exactly sure what the sources are, but this membrane permeability can potentially be a new quantitative in vivo functional contrast, but we need to still work a bit harder for that. Okay, so to wrap up, basically, I hope I've convinced you there is detectable ADC-fMRI contrast that is completely distinct from bold. And so the bold mitigation is challenging, but it's not impossible with some careful protocol design. And I do think this method has potential for white matter fMRI. At the level of task fMRI, we have good activation clusters in the white matter, particularly with isotropic encoding, and no detectable contribution from purely vascular challenges. So this is very encouraging. And in the rodent, there seems to be distinct signatures between primary cortical and subcortical areas. In the resting state, we have stronger white to white and white to gray connectivity with ADC-fMRI than we do with bold, and different graph metrics. We're just not sure yet what they mean exactly. And if we want to go beyond ADC, it seems to be a good avenue looking at this permeability fMRI, but for now, we've seen it in the rodent. We tried to do it in the human, but the sensitivity was not high enough. So this is still very much a work in progress. So I'd like to thank everyone in my group who contributed to this work, and of course, many collaborators across different sites. And well, thank you very much for the invitation again and for listening.
A great talk, Ileana. Thanks a lot. We learned a lot. So I see some questions in the chat. Marco, do you want to unmute yourself and ask?
Yeah, sure. Hold on. Yeah, so to go back to your slide on the dorsal and ventral stream, I noticed that your map was basically stopping at MTV5. Now, functional data seem to suggest that there are actually three streams. The classical ventral that goes all the way down to the inferior temporal cortex. The classical dorsal that goes all the way up to the posterior parietal cortex. And then this intermediate third stream that through MTB5 should go to the superior temporal cortex. So I was wondering how much your connectivity, your mapping can actually map all these connection all the way to say functional areas in the infratemporal cortex or functional areas on the superior temporal cortex and functional areas onto the posterior parietal cortex. Of course, I'm not talking about functional stuff. I'm talking about your mapping connectivity.
Yeah, thanks for the question. I don't think we've looked at this in much detail. So basically, what you're saying is that here, in addition to these areas, we should be looking at other areas that connect to this visual system. So I'm not exactly sure if they are represented here. And this experiment, unfortunately, was also not optimal in that regard, because it was still with the linear encoding, so we didn't have this isotropic sensitivity throughout the brain, and also to have a reasonable temporal resolution. So we went with a repetition time of 1 second. At the time, we also didn't have multiband available properly, and so this was partial brain coverage. So we had this slab, so we couldn't look at full brain coverage with this connectivity, but it would be definitely interesting to investigate. I think our isotropic ADC-fMRI data that came later with the visual stimulus does have full brain coverage. So then maybe we could follow up on this and look at other brain parcellations and how this connectivity looks with what you suggested. That's a great idea, actually. Thank you.
Sure.
Joseph Gorgi had a question in chat. Ileana, do you want to just read it?
Oh, sure. Cerebrovascular reactivity beyond hypercapnia with breath hold and rebreathing. How can this increase the sensitivity specificity for detecting pathology? I'm not sure I understand the question. So I guess here the goal, the idea with this experiment was to show that ADC-fMRI, if we really remove the T2 weighting and the sensitivity to background gradients, etc., we are actually not sensitive to this cerebrovascular reactivity. So we are not sensitive to purely vascular changes that do not have underlying neural activity. And so maybe what you're suggesting is that thanks to this, basically now we have a dissociation between the neural activity and any potential vascular impairment. And I think this is important for diseases where you have a bit of both, right? So often if we find say hypoactivity or hypoconnectivity in bold fMRI in, I don't know, multiple sclerosis or dementia, etc., we tend to interpret this as a change in brain activity, but it could also be that it's a purely vascular effect. And so I think using bold contrast and ADC-fMRI contrast in a complementary way can actually maybe better shed light into this pathology and impairment, whether it's a vascular problem causing the bold hypoconnectivity, or it's really neural activity that has kind of slowed down. I think this would be an interesting research lead. Yeah, I'm not sure if I answered the question, but
Then there was, okay. Then, yeah, looks like you answered his question. There was another basic question. What's the difference between ADC and DTI? So actually, those of you who don't work on the math behind how to compute tensors or compute these ADC values, maybe just like one sentence.
yeah, of course. And
This,
then maybe I should have clarified this.
yeah,
So DTI basically, so you acquire several directions in space, and then you reconstruct your diffusion behavior in this Gaussian approximation regime as an ellipsoid, where basically the longer axis would give you the principal direction of your diffusion, and then the rest is maybe more shrunk because you're perpendicular to your white matter fiber, etc. So this is used more for microstructure reconstructions or understanding how the tissue is laid out, particularly in white matter. And ADC, so apparent diffusion coefficient, is just really this diffusivity in one chosen direction of space. So with DTI, basically you can reconstruct your apparent diffusion coefficient in any direction. So you have your tensor, you project on that direction, you know what the ADC is in that direction. Here, basically we're choosing one direction to still have good temporal resolution. And then it just depended how that direction was basically lying with respect to the fiber orientation, which was problematic in terms of having good sensitivity everywhere. So in human research context, so if you're looking at microstructure without any sort of functional content, definitely DTI is really important because there is a lot of anisotropy in the human brain. From a functional perspective, you cannot afford to reconstruct a full tensor. You need too many directions. So either you do ADC-fMRI with one arbitrary direction, or you do this isotropic encoding that we are doing, where basically you're sensitizing to all directions in space at once.
great. This is a very nice explanation, Ileana. Thanks. Maybe I missed this. Let's see if there was another question. So, maybe I missed this. Did you say the source of the ADC signal was from neuronal swelling or these axonal deformations? Maybe I missed that part.
Yes, so well, this is what I suggested, but if you ask me explicitly, we also don't have a full answer, right? So basically, these papers showed where was I here? right? So these papers basically showed that this effect exists in neurons and
Hmm.
axons, but we cannot exclude that our source of contrast with ADC is also, for example, from astrocytes. Because astrocytes, they also have this very large ability, actually even more so than neurons, to take up water and then push it out again to maintain ion homeostasis. So they really need to maintain ionic imbalance. And I think they're even more morphologically flexible
Hmm.
than neurons and axons to some extent. So we don't know whether our contrast comes from neurons or astrocytes. We did some simulations on synthetic white matter substrates that really suggested that whether it's axons, sorry, oh yeah, whether axons or astrocyte swelling, the ADC drop really comes from extracellular water that becomes much more tortuous. So it's not so much the intracellular space, but it's really the fact that on the outside everything becomes harder to get around. So
Hmm.
this is really an extracellular effect, but we don't know if it's the neuron swelling, axon swelling, or astrocytes. It could be a combination. What we also know is that it's not vascular because we really tested it, and also the timings don't match. I mean, the vascular response is always heavily delayed, and we always measure responses that are really smack on the task timings. So yeah.
Ah, interesting. So then this has implications for interpreting the ADC-fMRI connectivity.
Oh, for sure. Yes. Yeah. Yeah. Definitely. Yeah. Yeah.
Okay. So I have a few more questions actually, but in the interest of time, we can thank Ileana for such a wonderful talk. Uh, and maybe Ileana, if you just want to stay here for a bit, maybe
Yeah.
I
Sure.
had a
Of
few
course. No
questions
problem. Yeah.
briefly.
Thank
Uh,
you, everyone.
so see everyone next time. Thanks.